So what if the reason a multi-million-dollar cancer immunotherapy fails isn't because of some massive whole-body breakdown, but actually because of a microscopic turf war?
Right, a turf war happening in a space that's, what, less than a third of a millimeter wide?
Exactly. I mean, it completely upends how we usually think about these things. We're
Yeah.
so used to this traditional systemic view of the immune system. But today, we're looking at a diagnostic landscape where hyperlocal cellular geography is the real key.
It's fascinating. And welcome, everyone, to today's deep dive. If you're curious about how these cutting-edge therapies actually work or why they sometimes don't, you are in the right place.
Yeah, we are on a mission today to uncover this microscopic mystery. We're talking about immune checkpoint blockade, or ICB. It's this incredible medical marvel that uses your body's own immune system to fight cancer. But, just doesn't work for everyone.
Which is the most frustrating part of oncology, honestly.
Right. So we're diving into exactly why that is by looking at these tiny little cellular neighborhoods. Our main source today is a really fantastic preview article in the journal Cancer Cell by Nicholas J. Salja and Jason B. Musich.
And they're reviewing a breakthrough study by researchers Hernandez-Verdin and their team, who managed to map out the tumor microenvironment using a mix of clinical, in vitro, and in vivo data.
It's some seriously heavy data, but don't worry, we're going to break it all down. So let's start with that phrase you just used, the tumor microenvironment.
Yeah, that term is really the core of this entire puzzle because, for decades, we've kind of treated cancer as this monolith. We assumed that if we pump the body full of immune-boosting drugs, the immune system will just flood the tumor and wipe it out.
Like a rising tide lifting all ships, right?
Exactly. But the clinical reality is just way more nuanced than that. We have patients who we call responders, whose tumors shrink dramatically, and then non-responders who sadly see no benefit at all.
And to figure out why, these researchers didn't just look at the patient's overall immune health in their blood or something. They zoomed way, way down.
Right down to the physical space where the cancer cells and the immune cells are literally touching each other.
Okay, but before we get into how the cancer actually sabotages the treatment, I feel like we need to understand the battlefield first. Like, how does the immune system even set up its defenses locally?
That is a great place to start. So the source material looks a lot at studies on renal cell carcinoma, so kidney cancer, to illustrate something called tertiary lymphoid structures, or TLS for short.
Tertiary lymphoid structures. Okay, so most of us know about lymph nodes, right? Like when you get a cold and your neck swells up.
Right, those are your systemic hubs. That's where immune cells gather, they train, and they launch their attacks from. But lymph nodes are usually geographically pretty distant from a solid tumor.
They're like the distant military academies.
Oh, I like that, yeah. Distant military academies. A TLS, on the other hand, is an immune hub that gets built on demand, directly inside or right next to the tumor itself.
Okay, let me unpack this with that analogy.
Yeah.
So if the lymph node is the military academy, the TLS is like a forward operating base.
Exactly. A forward operating base constructed right in the trenches. And what the clinical data shows is that in the responders, the patients who actually beat back the cancer with the immunotherapy, these forward operating bases are highly active.
So they're buzzing with activity. What does that actually look like biologically?
Well, there are two main things happening in a functional TLS. First, you see really high antigen presentation.
Meaning the immune cells, like dendritic cells and B cells, are basically capturing little pieces of dead cancer cells, right?
Right. They capture these fragments, these antigens, and hold them out on their surface to train the T cells. I always think of it as handing out the molecular barcodes of the enemy.
Handing out the barcodes. Got it. So that's the first thing. What's the second?
Well, because that barcode is being actively passed around, we see a massive influx of plasma cells moving into the TLS.
And plasma cells are just fully matured B cells, right?
Yep, exactly. And in a responder patient, these plasma cells are successfully pumping out high volumes of IgG antibodies, immunoglobulin G.
Okay, IgG. I know that's sort of the holy grail for fighting solid tumors, right?
It really is the primary workhorse, yeah. It has this specific shape that lets it bind super tightly to the tumor antigens. And once it's attached, the tail end of the IgG, the FTA region, acts like a glowing beacon.
Like painting a target for an airstrike.
Spot on. It recruits macrophages and natural killer cells to basically come over and eat the cancer cell.
Okay, so the forward operating base is built, the communication lines are open, and the troops are manufacturing these highly targeted IgG weapons. But hold on, here is my pushback.
Let's hear it.
If the immune system is perfectly capable of building this on-site base camp, and the plasma cells are stationed right there inside the tumor mass, Why are some patients still failing the therapy?
That is the million-dollar question.
Right, because the clinical data in the source shows that the physical architecture of the TLS is actually there in the non-responders too. The fort is built. So why are they losing the battle?
Well, that's where we get to the bad neighbor effect. Because just having the base camp there isn't enough. It turns out the neighborhood you build it in matters immensely.
Okay. Here's where it gets really interesting. Tell me about these bad neighbors.
So the researchers shifted their focus. Instead of just looking at the immune cells in the fort, they mapped the metabolic output of the cancer cells sitting right next door. And they found a specific subset of cancer cells in the non-responders, which they call PT-like cancer cells.
PT-like cancer cells. And these guys have a very distinct way of fueling themselves, right?
They do. The source notes they have a highly active TCA cycle.
Okay, let's pause. The TCA cycle, that's the tricarboxylic acid cycle, right?
Yeah.
Or the Krebs cycle, for anyone having high school biology flashbacks.
Yes, the Krebs cycle. It's how cells generate energy. But in these PT-like cancer cells, the cycle isn't just running fast to fuel tumor growth. It's actually being metabolically reprogrammed.
Reprogrammed how?
The cancer cells are literally siphoning off intermediate metabolites from the cycle. Specifically, they take something called alpha-ketoglutarate, and they convert it into glutamate.
Okay, glutamate.
And then they decarboxylate that glutamate to produce massive, massive amounts of GABA.
Wait, GABA, as in gamma-aminobutyric acid.
The very same.
But isn't GABA a brain chemical?
Yeah.
Like in neurology, GABA is the main inhibitory neurotransmitter in the human brain.
Right.
It calms the nervous system down.
Exactly. It binds to receptors and opens ion channels to reduce neuronal excitability. But we aren't in the brain here. We are in the tumor microenvironment.
Oh, wow. So the cancer cells are basically co-opting a brain chemical and using it as a weapon.
Yes. They've weaponized a neurotransmitter as an immunosuppressant. They're pumping this GABA out into the space around them. And when that GABA binds to the neighboring immune cells, it acts like a blanket of chemical static.
Chemical static.
Right. It hyperpolarizes the immune cells, which basically blocks the activation signals they desperately need to function.
Okay, but here is a really specific detail from the source data that blew my mind. The researchers pinpointed exactly how far this chemical static reaches.
The 300 micrometer radius.
Yes, the impact only happens within a rigid 300 micrometer danger zone between the cancer cells and the TLS. That is less than 1/3 of a millimeter. We're talking about the width of maybe 3 human hairs.
It is incredibly small.
So my question is, if this GABA production is so potent that it ruins the therapy, why doesn't it just shut down the patient's entire immune system? Why is this hyperlocal, super tiny distance the key to the whole mystery?
That really comes down to the biophysics of the tumor itself. You have to remember, tumors aren't just loose collections of cells floating around. They are dense, fibrotic masses.
Right, they're tough.
Very tough. And they have extremely high interstitial fluid pressure. So molecules like GABA don't just flow freely through them like dye in a glass of water. They have to push through a really dense matrix.
So the GABA gets diluted pretty quickly.
Exponentially quickly. As the cancer cells pump it out, the concentration drops off a cliff the further away it gets. The actual concentration required to paralyze an immune cell only exists within that 300 micrometer radius.
Oh man, so it's literally a short-range frequency jammer.
That is the perfect way to describe it. The cancer doesn't have to fight the whole systemic immune system. It just builds a house on the fence line of the TLS and blasts enough chemical static to blind the guards standing right outside its door.
That is deeply sinister and incredibly smart from an evolutionary perspective.
It really is. If a PT-like cancer cell is inside that 300 micrometer zone, the GABA just diffuses straight into the TLS, and the immune system's antigen presentation completely plummets.
The barcodes stop being shared.
Right. The barcodes aren't communicated, and the whole local response just collapses. It's a localized sanctuary for the tumor.
Okay, so knowing that this GABA cloud is the weapon, the source material then zooms in even closer, right? Because we need to know exactly how this jammer is dismantling the immune cells caught inside the cloud.
Yeah, so to figure this out, the researchers had to move away from the clinical biopsies of patients and use in vitro models. They had to isolate the variables in a lab.
So dish experiments.
Right. They took primary human B cells, which, remember, are the precursors to those plasma cells we want, and they cultured them in the lab. Then they stimulated them, giving them the activating signals you'd want during immunotherapy.
But they also dumped GABA into the dish to simulate the bad neighborhood.
Exactly. And the addition of the GABA triggered 3 very distinct crashes in the B cells.
Okay, let's go through them. The first one the study mentions is that B-cell proliferation just drops.
Yeah, it causes cell cycle arrest. Normally, when a B cell sees a tumor antigen, it's supposed to undergo clonal expansion.
Basically rapid cell division, right? Building an army of clones.
Right, to target that specific threat. But the GABA receptor engagement hits the brakes on the B cell's internal signaling. The cell cycle halts, and those reinforcements just never materialize.
No army. Okay. The second crash is a severe decrease in HLA-DR expression on the B cell surface. Now break down HLA-DR for us.
Sure. So HLA-DR is a type of MHC class II molecule.
Okay, tying back to the antigen presentation from earlier.
Exactly. HLA-DR is the physical little receptor arm that holds the fragmented tumor proteins on the outside of the B cell. It holds it up so the helper T cells can come by and inspect it.
So if HLA-DR expression drops,
the B cell literally loses the physical structure needed to present the tumor's barcode. The whole communication network between the B cells and the T cells is completely severed.
Wow. Okay, so proliferation is dead, communication is severed, and that brings us to the third crash. Overall immunoglobulin, or IgG, secretion plummets.
Right. Antibody production drops.
But wait, so what does this all mean when we look at the whole picture? Because here is where I saw an apparent contradiction in the data.
Oh, how so?
Well, the dish experiment says Ig secretion drops across the board. But when you look at the clinical graphics from the actual human patients, the non-responders' TLS isn't just producing nothing. It's actually producing a different antibody. They were making IgA instead of the helpful IgG.
Ah, yes.
So is the immune system suppressed, or is it just confused?
That is such a brilliant nuance to pick up on, and it's key to the whole study. The base camp hasn't been completely leveled. Its internal machinery has been corrupted.
Corrupted how? Like bad programming?
Kind of, yeah. A healthy anti-tumor response is what we call a Th1 phenotype. That's the programming that tells B cells to undergo class switching so they produce IgG.
The solid tumor killer.
Right. But the GABA disrupts that specific signaling pathway. So yes, the overall volume of immune activity is dampened because there are fewer cells and less HLA-DR, but the B cells that are still active get pushed onto this aberrant, twisted pathway.
And that leads them to make IgA.
Exactly.
But why is IgA so useless against a solid tumor compared to IgG? An antibody's an antibody, right?
Not quite. It's an issue of structural conformation. They have different biological jobs. IgA is really designed for mucosal immunity.
Mucosal, so like the gut.
The gut, the respiratory tracts, saliva, it's meant to neutralize pathogens before they break through the epithelial barrier. It actually often forms a dimer, basically two antibody units linked together.
Okay, so it's bulky.
Right, and while it's amazing at trapping bacteria in mucus, it doesn't really have the right structure to powerfully engage those FEC receptors on the macrophages deep in tissue.
Ah, it's a mismatched key.
Exactly, a mismatched key. The TLS is churning out these antibodies that are structurally completely incompatible with the locks required to destroy a solid tumor.
That is wild. The forward operating base is being tricked into manufacturing, like mucosal defense weapons, while it's actively fighting a deep tissue ground war.
That's a great way to put it, and that's why the immune checkpoint blockade fails. It's trying to unleash an immune response that has already been fundamentally reprogrammed by the cancer's local environment.
Okay, so if we know the mechanism, if GABA is the jammer and it's making the B cells manufacture mismatched keys, then how do we fix it? How do we shut down the jammer and save the TLS?
Well, that realization led the researchers to test a counterattack in living systems. They moved to their in vivo animal models.
Right, the study used a soft tissue sarcoma model in mice. And the source mentioned that sarcomas are really useful here because they naturally develop these tertiary lymphoid structures, right?
Yes, they are a fantastic proxy for the immune architecture we see in the human patients. So, to counter the GABA defense, the researchers decided to test a combination therapy.
2 drugs at once.
Exactly. They used an immune checkpoint blocker, specifically alpha-PD-1, alongside a GABA inhibitor.
Okay, wait. Let's talk about why they did a combo. Because if alpha-PD-1 is the standard immunotherapy, and it wasn't working before because of the GABA, why do we still need it?
That's a really logical question.
Like, why not just give the GABA inhibitor alone, drop the static, and let the immune system do its thing?
Because inhibiting the GABA only solves half the problem. You clear the static, sure, but you don't strip the tumor of its other native defenses. Let's break down how the PD-1 drug works.
Okay.
PD-1 is a receptor on T cells, and cancer cells often express the matching ligand, PD-L1. When the cancer cell's PD-L1 touches the T cell's PD-1, it sends a "don't eat me" signal.
It just exhausts the T cell.
Right. So the alpha PD-1 drug blocks that handshake, which theoretically unleashes the T cell to attack.
But in the non-responders, the alpha PD-1 fails because the T cells never even get the barcode in the first place, thanks to the GABA cloud.
Exactly. But if you only give the GABA inhibitor, here's what happens. The TLS wakes up, the B cells start proliferating again, HLA-DR comes back, they switch back to making the right IgG antibodies.
So the base cancer's fixed.
It is, and the T cells get recruited. But when those T cells finally arrive at the tumor surface, they run smack into that PD-L1 "don't eat me" signal.
And they just get exhausted all over again.
Precisely. You have to solve both localized problems simultaneously. The synergy between the two drugs is completely non-negotiable.
You drop the cancer's chemical shield with the GABA inhibitor, so the base camp comes online, and then you use the immunotherapy to remove the final brake on the T cells.
That's exactly it. And the in vivo data strongly supported this. The combination of the GABA inhibitor and the alpha PD-1 drastically decreased tumor growth in the mice.
And it didn't just shrink the tumor, right? The spatial analysis in the source showed that it literally reshaped the microenvironment.
It did. It actively increased both the T cells infiltrating the area and the presence of those TLS resident B cells. By understanding the biophysics of how the GABA diffuses and the metabolic shunting of the Krebs cycle, the researchers basically engineered a way to manipulate the microgeography of the disease.
They didn't just blindly boost the systemic immune system, they went in and repaired the 300 micrometer dead zones.
Brought them right back online.
You know, taking a step back, this represents a massive shift in how we view disease resistance, because we usually frame clinical failures through this macro lens, right?
We do. We ask, why did the patient's body reject the drug? Or why is their immune system so weak?
Exactly. But this deep dive really drives home that biological failures aren't always systemic. Sometimes the reason a miracle cure fails is just a hyperlocal neighborhood dispute at the cellular level.
It's the failure of a specific 300 micrometer radius where the cancer managed to build a sanctuary.
It just changes everything about how you think about medicine.
The implications for biotech and drug development are truly massive. We are moving past the era of treating tumors as these uniform, homogeneous lumps. We're entering the era of spatial pharmacology.
Spatial pharmacology, I love that term.
We have to map the physical coordinates of the immune cells. We have to analyze the specific chemicals being exchanged in those microscopic spaces, and then tailor our drugs to clear those very localized blockades.
Which really leaves us with one incredibly provocative final thought for you listening at home to mull over. We now know that these cancer cells can metabolically reprogram themselves to co-opt an everyday neurotransmitter like GABA and weaponize it to create a 300 micrometer force field.
Right.
If a tumor can achieve that level of chemical engineering with just GABA, what other invisible, highly localized microclimates are tumors constructing right now to hide from our best medicines?
It's a chilling thought, honestly. The metabolic flexibility of cancer is arguably its greatest survival tool. Every intermediate byproduct of cellular respiration could potentially be a weapon if it's secreted in the right concentration, right next to an immune hub.
The diagnostic waters are undeniably complex. It demands that we look closer, that we measure the physical distances between these cells and figure out how to decode the chemical static that's jamming our defenses. Thank you so much for joining us on this deep dive into the microscopic battlegrounds of immunotherapy. We'll see you next time.